Hi everyone, my name is Raneet Kahlon. Thank you so much for joining me today as I share a little bit about my summer research in the Dr. Carol Huang lab. Uncovering the role of the prolactin receptor in glucose homeostasis beyond pregnancy. My sort of organ of interest, if you will, over the summer was the pancreas, in particular the beta cells within the pancreas that are responsible for secreting insulin, which reduces blood glucose levels. On the right here, I have an immunofluorescent image that I took over the summer of the pancreas. The structure in the middle that's insulin positive in the green is the islet, and these blue dots within it represent the beta cell nuclei. Now we know from previous literature that the body has to adapt to metabolic stresses like pregnancy, obesity, and high-fat diet. And one of the ways in which this adaptation occurs is by increasing beta cell function. And I want to highlight one particular mechanism, and that's the incretin effect. Incretin hormones, GLP-1 and GIP, are released from the gut after eating a meal, so they're subject to that oral pathway, and they travel to the beta cells to potentiate up to 50% of all insulin secretion, so really crucial mechanism here. And we know from previous research in the Huang lab that the prolactin receptor is really important for this adaptation to occur during pregnancy. But with new clinical data, the question remains, what is its role of the prolactin receptor outside of pregnancy? And that's how my research question was born. What is the role of prolactin signaling in pancreatic islets beyond pregnancy? To answer this question, our experimental design looked a little bit like this. We used beta cell specific inducible homozygous prolactin receptor deletion mice. Here and out, I'll refer to these as the knockout mice. This basically just means that both functioning copies of the prolactin receptor in these mice in the beta cells have been removed, so they don't really have a functioning prolactin receptor in the beta cells. And these were compared to their corresponding wild-type controls, which did have the functioning prolactin receptor. And we studied these mice after 2 pregnancies and 12 weeks of high-fat diet, and this was to really simulate the experience of mothers adopting a Western diet that's high in fats and sugars postpartum. So the first thing we saw is an impaired insulin secretion ability in the oral glucose tolerance test in the knockout mice depicted here in the orange. So in the oral glucose tolerance test, we give glucose bolus directly into the mouth that travels through the oral pathway and is therefore subject to that incretin effect I had mentioned earlier. So we can see here that the orange knockout mice have severe impairment of insulin secretion ability compared to their blue wild-type controls. But if we compare this to the intraperitoneal glucose tolerance test that bypasses the incretin effect by directly injecting glucose into the bloodstream via an IP injection, we no longer see that impairment in the knockout mice. So this suggests that there is an impaired incretin effect in these knockout mice that are missing the prolactin receptor. So we wanted to investigate this a little bit further, and so we employed a qPCR technique to look at the incretin hormone receptor mRNA expression. So we looked at GIPR receptor expression and GLP-1R receptor expression, and you can see that the knockout mice in the orange have significantly less mRNA of both of these receptors compared to their wild-type controls. We went a step further and also looked at various genes that are known to regulate GIPR and GLP-1R expression, so E2F1, FOXO1, PAX6, PPAR-gamma, and TCFL2, and each of these genes are also downregulated in the knockout mice compared to their wild-type controls. So in summary, we've seen that these knockout mice, which are lacking the functioning prolactin receptor in their beta cells, they have an impaired incretin effect, which is a really important mechanism of insulin secretion. Further supporting this conclusion, they had reduced expression of incretin hormone receptors, so that was at GLP-1R and GIPR, elucidated through the qPCR data, as well as reduced expression of genes that are known to control incretin hormone receptor expression. So if you look at this figure here on the right, you can see that we have identified GLP-1R and GIPR as novel targets of prolactin receptor signaling, perhaps with these genes acting as intermediary steps. So a really interesting pathway to uncover. Finally, I'd like to leave you with the significance of this work. It's so important to study both gestational and type 2 diabetes in context of each other, because over 50% of women with gestational diabetes progress type 2 diabetes later in life. And so our project really moves us closer to defining prolactin receptor as a risk factor and hopefully being able to implement this knowledge into therapeutic targets in the future. Thank you very much. Please let me know if you have any questions.